Hormones are messengers sent by one cell to tell another cell to act or stop acting. However, hydrophilic or water-soluble hormones cannot cross the phospholipid bilayer of the cell membrane. So the receptors for these hormones exist as transmembrane proteins embedded in the cell membrane. When the hormone binds to the protein, a series of reactions is initiated that will eventually lead the cell to carry out the desired effects of the hormone. One of the chemicals involved is called a second messenger. The hormone itself is the first messenger. Let's look at an example of this process step by step. The hormone binds to the receptor on the outside of the cell. This new complex activates a G protein on the cytosol side of the plasma membrane. The active G protein then activates an enzyme that is an integral membrane protein called adenylate cyclase. Adenylate cyclase catalyzes a reaction that removes two phosphate groups from adenosine triphosphate, or ATP. The product of this is adenosine monophosphate, or cyclic AMP. Cyclic AMP is known as our second messenger. Cyclic AMP activates enzymes called protein kinases, which are enzymes that modify other enzymes by adding phosphate groups to them. This is called phosphorylation. The active protein kinases start phosphorylating enzymes, activating or deactivating them. These enzymes are now responsible for carrying out the various metabolic effects desired by the hormone in question. For example, glucagon is a hormone that raises blood sugar levels. The enzymes will result in the breakdown of glycogen, the storage form of glucose, into the bloodstream. All this time, an enzyme called phosphodiesterase is inactivating cyclic AMP. This way, the cell only undergoes this process as long as the hormone is present. You don't want the cell to keep doing this if your body doesn't want it to.